All right, let's get right into it. Today, we are cracking open one of the most incredible stories in biology, how a virus gets into our cells. But forget the boring textbook diagrams. We're going to treat this for what it really is, a high-stakes, microscopic heist. So here's the big question, right? How on earth does this tiny little package of code, something that isn't even technically alive, manage to break into a living cell, which is, let's be honest, one of the most complex and well-guarded factories in the known universe? Well, it's not by accident, that's for sure. This is a beautifully, or maybe terrifyingly, planned operation. And here's our master burglar, the SARS coronavirus. Its entire existence is built around one single goal: get inside that factory and take over the whole production line. But before we get to the actual break-in, you gotta understand the motive, right? Every great heist has a why, and that brings us to our first section, understanding the nature of this ultimate parasite. See, a virus is what's called an obligate parasite. That's a fancy way of saying it's completely helpless on its own. It's just a little packet of genetic material in a protein shell. It can't eat, it can't grow, it can't make copies of itself. It needs us. It has to hijack the machinery inside our cells to do its dirty work. And this isn't some rare, niche strategy. Oh, no. The source material points out that we know of at least 24 different families of viruses that cause disease in humans. That's a huge rogues gallery of burglars, and each one has its own special set of tools. Okay, so on to the next phase of the heist. A smart burglar doesn't just run up to a building and start smashing windows. They case the joint first. For a virus, that means finding the exact right door to get in. Now, these doors are actually specific molecules on the surface of our cells. We call them receptors. The best way to think about them is like a very specific kind of doorknob or a lock. And the virus, well, it's carrying the key. And this is that make or break moment. You can see the virus here, covered in these spike proteins. Those are the keys. It bumps up against a cell, which is covered in all sorts of receptors or locks. If the key doesn't fit the lock perfectly, game over. The virus literally just bounces off.
And this is so important, because this lock and key system determines everything. It's the reason a flu virus goes for your lung cells and not, say, your muscle cells. The locks are different. It's also why some viruses can only infect bats, while others can infect humans. It all comes down to having the right key for the right door.
All right, the virus has cased the joint, it's found the right door, and the key fits the lock. Now for the action sequence. It's time for the great break-in. And when it comes to getting past the security, viruses basically have two main plays in their playbook. The first is what we call direct fusion. Think of this as the brute force method, basically melting right through the wall. The other is endocytosis. This is the sneaky Trojan horse tactic. So with direct fusion, the oily outer coat of the virus literally merges with the oily outer membrane of our cell. They're made of similar stuff, so they can just fuse together, becoming one. And just like that, the virus dumps its genetic cargo right inside. HIV is a classic example of a virus that does this. Endocytosis is way more cunning. The virus just latches onto the outside of the cell. And the cell, thinking it's snagged a piece of food or something useful, does the work for it. It wraps a piece of its own membrane around the virus and pulls the whole thing inside in a little bubble. The virus has passed the wall, and it got the cell to be its getaway driver. Influenza is an absolute master of this move. So, we've seen what they do, but the how is where things get really wild. How does a tiny protein actually pry open a cell membrane? Well, it all comes down to this incredible piece of molecular machinery, the shape-shifting master key. Take a look at this. This is a model of the flu virus's entry protein, a famous one called hemagglutinin. These things are like the Swiss Army knives of invasion. They do it all. They act as the key to find the lock, and they're the crowbar that forces the door open. To understand how it works, we have to talk of physics for a second. These proteins are built in what's called a metastable state. Just think of a mousetrap that's been set, or a spring that's been compressed all the way down. It's packed with potential energy, just sitting there, waiting for something to trigger it. And here's how that mousetrap gets sprung. First, you have the protein in that tense, high-energy shape. Then, a trigger gets pulled. For the flu virus inside that Trojan horse bubble, the trigger is the acid inside the bubble. And then, snap. The protein violently changes its shape, snapping into a new, much more stable form. All that stored up energy is released at once, and it's that physical force that literally punches a hole in the bubble membrane, setting the virus free inside the cell. The heist is complete. Now, here's the cool part. Once you know every single step of the heist, you can start figuring out how to stop it. And that brings us to our final section, how we can block the entry. You know, the author of our source material, Dmitry Dimitrov, really nails it with this quote. He basically says that the more we learn about this break-in, the more chances we have to develop new ways to fight it. It makes perfect sense, right? If you know how the burglar picks the lock, you can build a better lock. And this is our toolkit for the counter heist. We can design drugs that literally sit on our cell's receptors, essentially gumming up the lock so the virus's key won't fit. Or we can develop antivirals that latch onto the virus's own entry proteins, jamming the key before it even gets to the door. And of course, the big one, we can use vaccines to train our body's own security guards, the immune system, to recognize these entry proteins and take out the virus on sight. But this is the thing that really keeps scientists up at night. This isn't a static fight. Viruses are constantly evolving, changing their keys, trying new ways to get in. So it leaves us with this one huge lingering question. In a biological arms race that's been going on for millions of years, how do we, as scientists and as a society, make sure we stay one step ahead?